Good morning. My name is Ben Busby. I'm the Genomics Outreach Coordinator for NCBI, and I'd like to introduce you to the NCBI Assembly Database. We'll have a closed caption recording of this video on our YouTube channel. You can see the URL below, and please subscribe to our YouTube channel. Tell your friends. If your question does not get answered, please either email webinars@ncbi.nlm.nih.gov, and if there are common questions we can't get to for whatever reason, we'll post the answers, those questions and their answers on our FTP site. Great. All right. So today I'm going to talk about obtaining genome sequences and annotations with NCBI Assembly. In my personal view, right now, Assembly is the fastest, easiest way to get genome sequences and annotations from NCBI, and I'll talk about why that is over the next few slides. First, I want to talk about the new Human Genome Assembly, GRCH38. GRCH38 fixes almost 1,000 loci from GRCH37, and there are about 178 alternate loci. These are particularly important for immunologically relevant regions of the human genome. For example, if you're talking about some sort of immunoglobulin locus, it does not make sense to have a human reference immunoglobulin locus, and that's why we have alternate loci. So please consider using GRCH38 for your analyses in this realm. More details on the differences between 37 and 38, as well as mouse 37 versus 38, can be found at the Genome Reference Consortium website shown here. Just Google Genome Reference Consortium. If I go to Assembly and I Google Homo sapiens, what I will see is the latest patch for GRCH38, that's obviously P3. I can click on that and then go into the FTP site. I'll show you how to bridge those two things in just a couple of minutes. And what I see here is I can grab the nucleotide FASTA, the GFF3 annotations. By the way, we're going to be releasing GTF annotations probably in the next 6 to 9 months. I can also see protein sequences as well as RNA sequences and their annotations. Many people also want to get sequences from other organisms, particularly bacteria. So we have about 20,000 bacterial genomes that are available for download in RefSeq and about 30,000 available for download in GenBank. Real quick, for those of you who are somewhat new to this, the big difference between GenBank and RefSeq here, GenBank are sequences and annotations that have been submitted by the user community. We do not adulterate them in any way. RefSeq, we typically don't change the sequences much for genomes, except maybe to put in mitochondrial sequences. That's for the pure genomes. For proteins, things can change a little bit. But sometimes we do put in new annotations based on our annotation pipelines, which I will show you in a little bit. So one might think you would want to go to Genomes at NCBI to download genome sequences, but what the Genome Database does right now is it shows genomes in their genera context with respect to each other. So what I have here is, and actually more likely species context with respect to each other. Here I have the several thousand Mycobacterium tuberculosis species for which we have sequences. And you can see a tree here with obviously quite a few of the nodes collapsed substantially. Our intention over the next 9 months to a year is to make this tree totally interactive, going down to the individual strain level from which you will be able to link to Assembly. But we have not enabled that functionality yet. Please be on the lookout for a webinar where we will actually solicit user input on how that resource should be laid out. Right now, like I said, I think the best place to get sequences for individual strains is from the Assembly database. So if I search Assembly for Mycobacterium tuberculosis, I get the very well studied, for example, H37RV strain up at the top. If I want to look at all of the H37RV strains we possess, I can certainly do that in the assembly database, and we can see where they are submitted from and when they were submitted. If I go to an individual H37RV record, the really nice thing is these two links on the right to the GenBank and RefSeq FTP sites. I apologize that this slide got a bit distorted. And then I can go and I can, if I click on that link, particularly for RefSeq, I can get the FASTA, I can get the annotations, protein FASTA, and annotations. Now, the thing we've done with the FASTA, and I'm very proud to say we've done this, is we have changed our genomic FASTA files such that they will work with popular RNA-seq analysis software such as Bowtie, HiSat, and STAR. So if you index these genomes for Bowtie, HiSat, or STAR, and then you use cufflinks, cuffdiff, or DE-seq, you will get actual expression, transcript expression values or gene expression values, depending on what you're trying to do. So that's a fairly useful thing to know, I think, right now. One thing to remind you guys who are doing that kind of work is that remember the accession in the FASTA has to match exactly with the accession in the GFF3 or GTF file. So right now, I would suggest not mixing annotations from UCSC or Ensemble with genomes from NCBI, although we do intend to make a converter in the near future. Also, if things have not been submitted as full genomes, if they've been submitted just as contigs, you can get those out of the nucleotide database. The easiest way to dump those sequences is to go to the Send To button. This is an often underestimated button in the NCBI Pantheon. If you go to the Send To button, you can send them to a FASTA file, as shown here. I wanted to talk a little bit about our genome annotation pipelines. These are user questions I get quite a bit. And if you have other questions about our annotation pipelines, please email us at webinars at ncbi.nlm.nih.gov. This is our prokaryotic annotation pipeline. If you submit us bacterial genomes as contigs, scaffolds, or preferably as whole chromosomes, we will annotate your bacteria for you. Also, we have a eukaryotic annotation pipeline. That's something that's obviously quite a bit more complex, and you can read very extensive documentation about that at this URL shown here. Also, this is a basic diagram showing that we essentially throw the kitchen sink at these annotations. These are annotations based on perceived need from the community. The ranking of these annotations is based on perceived need for the community. So if you believe that there is a genome that really needs to be annotated by NCBI, please email genomes@ncbi.nlm.nih.gov and we will consider your request. If you have any other questions about eukaryotic genome annotation, please email genomes@ncbi.nlm.nih.gov. I wanted to talk just a little bit about de novo RNA-seq assembly. We do house quite a bit of data in that realm, although not typically in mouse or human, but in non-model organisms, we have millions of records for de novo RNA-seq assembly, and those can be found in the transcription shotgun archive, which is part of the nucleotide database. I can talk about that more extensively if anybody has questions. Finally, I would like to thank a large amount of people for their contributions to these databases, as well as their help with putting together this presentation. And for the next 5 to 10 minutes or so, I'll be available to take any questions. Thank you very much. I hope all of you have a fantastic day. And for those of you on the East Coast, welcome to spring. Thank you. All right, so this is a pretty good question. As a rank beginner to assembly, where would you suggest I begin? What I would do is go to the assembly database, and I would dive in head first. I would go to the assembly database, and I would look for your genome of interest, and I would try to download the FASTA file and do whatever it is you want to do. One thing, so I teach a lot of bioinformatics courses, and one thing I often tell my students is it is best to start with a biological question when you're doing computational things, because you are most likely a biologist. So do we have any assemblers on NCBI? We do not have assemblers on NCBI. It's a good question. There are a couple of websites that outline assemblers. I cannot endorse anybody's product, so I can't speak to pros and cons of different assemblers, but what I can say is omixtools.com is a pretty good place to look for sort of just a listing of tools for this type of thing, including assemblers. Some popular ones are Velvet, Soap de Novo, Abyss. There's a bunch of them. Also, Broad has an assembler comparer called MetAmos, so you might want to check that out. Can one download plasmid genomes? Yes, you can. We don't have a ton of them, and often they will come with whatever bacterial species that they were discovered with. So they will be listed separately in the FTP site. And some of my colleagues may wish to weigh in on that. That's something we've been talking about a lot. So if they have more, we'll put them in the FTP site linked from the video. That's a great question. If there are no other questions, oh, by the way, the NCBI, please subscribe to our YouTube channels. And by the way, the NCBI minute is going to change from 9:45. We're going to move it till noon to make things a bit easier for our friends on the West Coast. So the next NCBI minute will be May 6th. We'll actually talk about PubMed Commons. It will be fantastic, and it will be at noon Eastern Time. All right, well, thank you very much, everybody, and have a great day.